Alrighty, so we're going to try Unit 2 slides again because I can't seem to figure out technology. Anyway, Unit 2 it is cell structure and function, and it is chapters 6 and 7 in the Campbell biology book. There are 136 slides, so I'll probably talk pretty quickly and then slow down, speed up as I need to, but buckle up, it might be a little while. Alrighty, so this is the cell theory is what this slide covers, and so all cells come from other cells. Everything is made up of cells, and that they all descended from cells, and it's the smallest. You can't just go poof and then there's a cell. You have to have a precursor cell. So that's what this is talking about. It's called the cell theory. This is just a picture. We will focus mostly on the stuff that can be seen by a light microscope and right around that area. We talk a little bit about organelles and stuff like that, but this class is a little bit about subcellular components or organelles and stuff like that, but a lot of it is how those different things interact and how we send signals through our body and different things like that. Alrighty, so there are two types of cells, prokaryotes and eukaryotes. And then there's big things called domains, and so there's three big groups: bacteria, which is what you think of as bacteria that make you sick and stuff like that; archaea, which sometimes is referred to as archaebacteria. It is a group of prokaryotes, a group of single-celled organisms that also share characteristics of eukaryotes, and then the third one is eukaryotes. And so there's single-celled things and multi-celled things, and it's a big different things fall under different categories, but there are all bacteria are prokaryotes, and then everything else is eukaryotes. So protists, fungi, animals, and plants, they are eukaryotes. And so every single cell, whether it's a prokaryote or eukaryote, has four things in common. It has a plasma membrane, which means it has a barrier that it holds so that everything gets held in, kind of like the outside of a balloon, but we'll talk about why it's semi-permeable later when we get to the cell membrane part. Some sort of liquidy part inside of it, it's referred to as the cytosol. Some kind of genetic material. Your book says chromosomes. I don't really love that because bacteria have genetic material, but they don't have chromosomes. So that's just me being picky. And then ribosomes, because every living thing requires some sort of proteins or enzymes in order for their metabolic functions to work. So prokaryotes are, like I said, bacteria. They are single-celled. What differentiates them from eukaryotes is they do not have membrane-bound organelles. So it means they don't have a nucleus, they don't have mitochondria, they don't have all the organelles that you might have learned about in middle school or maybe your biology class before you got to AP. But there's no nucleus, but there is genetic material. It gets stuck into a little region called the nucleoid region, or the nucleoid, so that when it goes through binary fission, which is how bacteria reproduce, that it pinches in the middle and then it becomes two. The nucleoid region is sort of a part where they get stuck, and so then it ensures that there's genetic material in both of the offspring, rather than if they were sort of both over here in sort of an oblong thing and we split it down the middle, this would have doubled the amount, this would have none of the amount, and that doesn't make sense. And so nature has it so that it has a little region where it kind of gets located. Cytoplasm, which is similar to cytosol. Cytosol is strictly the liquidy part. Cytoplasm is the liquidy part plus the organelles or plus some chunks. So maybe it's genetic material, maybe it's ribosomes, but it's just a slightly different word. And then just a picture of a generalized bacteria. This is called a rod shape. Sometimes you might see it called a bacillus, which is over on the right-hand side. And so strep, if you've ever heard of strep, no, not strep, E. coli, that is a rod-shaped bacteria. And then eukaryotes are, EU means like true or typical. So eukaryotes have membrane-bound organelles. They have DNA inside a nucleus. And then inside the nucleus is called nucleoplasm, and then from the nucleus out to the plasma membrane, that's called cytoplasm. They're usually bigger than prokaryotes, but they're still really, really small. The plasma membrane, which we will talk about at the end of this unit, is selectively permeable. It has different polarities, hydrophobic, hydrophilic parts, and so only some stuff gets through. Some stuff gets through super easy. Some needs a little bit of a helper, but like I said, we'll talk about that more later. And so then we have this little picture, and this just shows us how it has polar heads and non-polar tails, or hydrophilic heads and hydrophobic tails. And then this just shows how sometimes there's some proteins shoved in there. And so it's just structurally, it's pretty unique. And so that gives us properties that a cell membrane can control things. And so one of the things that is an important thing about cells is they have to be small. And so we watched a video about how people like up where it's cold, they're like shorter and rounder, and then like on the equator, they're taller and skinnier. It has to do with surface area. It has to do with heat. It has to do with waste, nutrient absorption, and getting rid of wastes and oxygen and carbon dioxide in and out of a cell. The smaller it is, the better. It's more efficient. And then this just gives us a graphic about surface area to volume ratio. We did a lab with the agar cubes and the methylene blue, and then we did a bunch of measurements and we calculated, and the one that had the highest surface area to volume ratio was the one that was the best cell as far as efficiency in the transfer of materials. And then the higher the number difference, so the higher the surface area to volume ratio, the better. And then the lower it was sort of the worse it was. So if you look on this one, 6 versus 1.2, the smaller cell is better. And so, like I said before, eukaryotes have membrane-bound organelles, and then plants and animals are both eukaryotes. They are pretty similar, but then there's some key differences. And so this is a picture of an animal cell, just a generalized animal cell. Not every cell has a flagella, but there's one on the on this picture. And so it just shows you the different organelles. We know that it's an animal cell because it does not have a cell wall or a large central vacuole or chloroplast. This one does, and so this one is a plant. And so the nucleus has the DNA. Ribosomes take the information from the DNA and then take it out into the cytoplasm and get with a ribosome and then synthesize proteins. When you hear the word ribosome, I want you to think about proteins. If ribosomes are present, then proteins are being made wherever it is present. And so as we talk through the organelles, that'll make more sense. And so this is just talking about the nucleus specifically and how it has its own special little membrane. It is phospholipid bilayered, very similar to the plasma membrane or the cell membrane. The nuclear membrane has its own special little one, and it also has ribosomes on it because we need proteins made that are used within the nucleus to make certain things. And so this is just a pictures of the stuff. And so if you can see, there's ribosomes on there. There's also some endoplasmic reticulum, just gives us a little picture. And then there's pores on the outside of our nucleus as well, because things need to get out. When we get to protein synthesis, one of the things that needs to come out of the nucleus is the copy of the DNA called messenger RNA. So it has to have a little hole. That's why there's pores on the outside, just like your skin on the outside of a nucleus. And so this is chromatin and chromosomes, and this is just vocabulary. And so chromatin is what I think of as like loose spaghetti. And so our cells, when they go through mitosis, they have to condense the chromatin into chromosomes. And so chromatin and chromosomes are the same thing. So I use this in class as a little example. A rubber band is the chromatin, but if I take it and I sort of twist it and then I let it sort of condense on itself, it's the same stuff, but it's a condensed form. And so what happens to chromosomes, or what happens to the genetic material, the DNA, is when it's ready to go through mitosis, it uses the energy to condense. And so it doesn't do that normally because it takes energy to do it. And so same stuff, different form, because it does it when it needs to. And then the nucleolus is a little dense spot. It's where ribosomal RNA is made. And so it's dense because there's a lot of proteins, and then that comes out and then it makes the ribosomes. out into the, sometimes they're on the nucleus, sometimes they're on the endoplasmic reticulum, sometimes they're out free floating within the cytoplasm. So that's where they come from. And so ribosomes are little, what I think of as like blobs, kind of like an anchor. And they have two little lobes, and then the messenger RNA hooks on, and then they are the site of protein synthesis. And so, like I said before, when we talk about ribosomes, we're going to talk about proteins. And so they just look like that. And then the endomembrane system, endo means inside, so it's the membranes inside of the cell, and it forms what's called the endomembrane system. So we have the outside of the nucleus, which is the nuclear envelope, then that makes a continuous system into the rough ER, then the smooth ER, then the Golgi, then it pops off with some vesicles or vacuoles, and then it can scooch out into the plasma membrane. The structure of all of those things is a lipid bilayer. And so what happens is, sort of big picture, is when I have my membrane and I form different things, and then I have a vesicle that pops off, and then I go to my, let me try and get my hand right, goes to my plasma membrane, and then it opens up and squirts the stuff out of the cell. It's sort of, it can just fuse with it because it's the same thing. And so it's structurally the same thing, so that there's no need for any sort of energy. It just sort of squishes up against it and then blends in. It sort of just becomes part of the plasma membrane. And so the endoplasmic reticulum, like I said before, there's two kinds. There's rough and there's smooth. The rough contains ribosomes, the smooth does not, because in the rough ER, proteins are synthesized. They are not synthesized in the smooth ER. There's different functions for that. And so this is just a picture. And then the smooth ER, rather than producing proteins, which happens in the rough ER because it has ribosomes, it has a couple of other functions. And so it makes lipids. Lipids are super important in our body for brain development, for insulation, for making sure that our joints don't get too like rough and squishy, or like it's just a nice, has a function. It gets bad rap because it's fat. It makes carbohydrates, it gets rid of poisons, and it stores calcium. We watched a video that talked about that if you're an alcoholic, hopefully not, then your body makes more smooth ER. And so if you drink a lot of alcohol, you make more smooth ER, so then you can drink more alcohol. So if you've ever heard of somebody having like a high tolerance, and so they have to drink more and more and more, it's because their body is accustomed to getting a lot of alcohol, so it has to get rid of those toxins, and so it made the machinery to get rid of it. And so then you have to, it's just a cool thing that our body does. Another example, kind of the same, in the same sort of thought process, is you make more of what you need where you need it. And so if you drink a lot of alcohol, you make more of the organelle that takes care of toxins. If you have muscles, which everybody does, in those muscles there are more mitochondria because muscles require more energy than do other kinds of cells. And so this is just talking about different parts and how they sort of make little packages. And so they make a little product, and then we send it down, and then we make it a little bit more perfect, and then we package it up. So it goes through this little process of things. So that's just what these next few slides are explaining. And so then the Golgi, and then this, as this talks about at the end, it says sorts and packages materials into transport vesicles. So we go through the rough ER, the smooth ER, the Golgi, and then a little bubble pops off, and that's called a vesicle. And so this is just a picture. And then a special kind of vesicles called a lysosome. Remember, lyse means to break open or to break apart. If it is a lysosome, it is a little vesicle that contains things that break something apart. So it contains enzymes, digestive enzymes. So if we eat, or if we have like cellular food that comes in and we bring it in, and then it sort of links up, I'll show you a picture in a second, with a lysosome, then they blend together, and the enzymes from one and the food from the other go together, and then we digest it, and then we get pieces and parts for different things that we need. And so when we need to eat, when our cells need to eat, it's called phagocytosis. And so if you are nerdy and you watch things about white blood cells, one of the white blood cells is called a macrophage. And so macro means big, phage means to eat, so it literally means big eater. So it goes around and just eats stuff that's not supposed to be in our body, bacteria or different stuff like that. So phagocytes are cells that eat, and then phagocytosis is the process by which cells eat. So if you break the words down, that's just what they mean. And so what happens is sometimes the little cell membrane or the plasma membrane opens up and then stuff comes into it. What usually happens is the membrane sort of goes and it wraps around a piece of something and then pulls it in and then links it up with a lysosome and then dissolves it. And so this is just a picture. And a little bit closer because I forgot to delete it. Same thing. And then one of the differences between an animal cell and a plant cell is that plant cells have a large vacuole. It's usually called the central vacuole. And what that primarily does is holds water. And so we have our brains say, hey, you're thirsty, drink some water. Plants don't have that. So they have to have this big like bubble of water. And so what that does is if you've ever had like a limp plant and then you water it and it perks back up, you're filling up its central vacuole with water, and then it makes it so that the cells are rigid and so then it can stand up. And so a few other kinds of vacuoles are just exactly what they sound like. So food vacuoles is when we do our little cellular eating, we make a little bubble with the food in it. Contractile vacuoles is protists are little single-celled organisms. They're cool. Some of them move by using contractile vacuoles. And so they sort of suck in some water and then squeeze it and shoot it out and makes like a little jet propulsion. But they contract it, and so it's exactly what it sounds like. And then a central vacuole, like I said, primarily holds water in plants. And so this is just a picture. And then the endomembrane system is different probably than you may have learned in previous classes. Hopefully not, but probably. But it is a system of membranes, and then it gives us like a pathway for something to follow in order to make and get better and get packaged and get to where it needs to be and go outside the cell and different stuff like that. And so just a picture. And then there are cool organelles that are thought to be sort of hijacked from in sort of pre-cellular times, and there's a bunch of pieces of evidence, but so one of them is mitochondria. The other one is chloroplasts. And so mitochondria is a site of protein, not site of protein synthesis. It is the powerhouse of the cell. It is the site of ATP synthesis. And then chloroplasts are where we absorb the sunlight and then we make glucose. And then peroxisomes are a cool little, extra little organelle that help us get rid of byproducts. But I'll talk about all of them. Cool thing, because I don't want to forget to talk about it later, is one of the big pieces of evidence that mitochondria and chloroplasts used to be outside of a cell is they both have their own DNA. And so there's mitochondrial DNA, there's chloroplast DNA. And so the mitochondrial DNA, I explained this in class, is if you think of an egg and a sperm, if you have an egg, has cytoplasm, it has genetic material, a whole bunch of stuff. A sperm's real small. All it has is genetic material. It doesn't have organelles or cytoplasm in a significant amount. And so when they blend to make an embryo, all the cellular components are from your mother. And so every single person's mitochondrial DNA, because it is a organelle that is in the cytoplasm, I'm not talking about DNA like your mom's DNA and your dad's DNA. Every single person's mitochondrial DNA came from your mother. And so my husband and I got 23andMe done, and you can trace, every single female is like 0.01 at least sub-Saharan African. And so we got 23andMe done, and mine said that I was 0.01 sub-Saharan African, and my husband's didn't have that. And I have that, and mine says that because I have mitochondrial DNA from Lucy. And if you don't know who Lucy is, do a little bit of research. And then chloroplasts are the same sort of thing. They have their own DNA. And so this is just talking through those a little bit. It's a lot of what I talked about. They're not part of that endomembrane system. They're not part of a path of things. They're sort of on their own out there. And then they have their own special little factories. It's really, really cool. And then I talked about the DNA. And so mitochondria and chloroplasts both have double membranes. And so we have to, when we talk about photosynthesis and cellular respiration, that will become significant. We have to have the structural ability to build up a gradient. And so that's why there's an extra membrane there. Just remember that it's there. We'll talk about it more when we get to the next unit. And then this just shows a picture, and then there's those weird folds. That's to give us a double membrane so that we can do cellular respiration. And then chloroplasts are, there's a bunch of different plastids. The famous one is chloroplasts. The way that I remember the difference between chloroplasts and chlorophyll is plasts sounds like plastic, and so the chloroplast is a container, and then chlorophyll is what fills it up. And I also remember Phil likes to color. He likes to color green, and so if you can remember that plastids hold it, chlorophyll is what's inside, and then the pigments absorb the light, which then we can do photosynthesis. And so the structures that are in this one is thylakoids, which is little stacks of green, looks like little green coins, and we can call them thylakoids. And then there's sort of some intermembrane, got a double membrane in the chloroplast as well. And so that fluid's called stroma. And so just a picture. And then peroxisomes, our body, by sort of how we go through our enzymatic processes, a byproduct that a lot of them make is peroxide. If you ever put peroxide on a cut, wouldn't be great if we just released that into our body. And so we have to have a way to get rid of that. And so peroxisomes are the little organelle that take care of our sort of harmful waste product. And then it just turns it into water, which is kind of handy. And so just a picture. And then this part's talking about structural things, like stringy structural things. And there's three that I want you to remember: microtubules, microfilaments, and intermediate filaments. To me, tubules and filaments, if I think about those two words, filaments sound smaller, and they are. And then so that means tubules are bigger, and then intermediate means it's between those two, which is exactly what it is. And so just a picture. And then one of the things that the microtubules, one of the functions of a specific kind of microtubule is where the motor proteins track along. And so if we think about, we made some proteins and we made a little vesicle, and it needs to get from sort of popped off the Golgi apparatus and needs to get to the plasma membrane. It doesn't just sort of go wherever it wants to. It gets a little track, and then it sort of hops. It goes like along it. And so that little thing is a tubule. And so it's a really cool thing. Why does it do that? Because it's more efficient than just going around and willy-nilly trying to get to the plasma membrane. And so this just shows a picture. Up at the top, it's just a picture. If you Google motor protein GIF, it's one, and it has this little thing that sort of travels along. That might help you sort of visualize it as well. And so then this is just talking about what I talked about before, sort of the size of those. And then microtubules are the biggest. They have different functions, sort of the structural rigidity of a cell to sort of like scaffolding to sort of hold it out. One other big thing, you probably called them spindle fibers if you labeled those in previous classes, is when we line up our chromosomes during mitosis, and then they sort of rip apart and they go from one side to the other. Then they're pulled by a little string, and those little strings are microtubules. Alrighty, and then this is just talking about sort of structurally where those tubules might sort of be generated from. And so centrosomes and centrioles, they're just structures, and then we'll look at some pictures. And so they look like this. And so centrioles are at the poles of the cells, and that's where those sort of the tubules come out of, and they grab onto the chromosomes. And then two special kinds of tubules, or like this is sort of like hair and tails on usually bacterial cells, sometimes protists, is cilia and flagella. And so cilia is little, like it's like hairy all the way around. Some protists have like a mouth hole, and so then the cilia sort of, they're usually in the water. They beat their cilia, and then it sort of makes a little current, and then the food goes into their little mouth hole. And then a flagella is like a big old whip tail, kind of like on a sperm. So some pictures just to show you what those look like. And then microfilaments are, for some weird reason, the AP likes to ask about which one's made of actin subunits, and that's microfilaments. And so filaments and tubules, contains an A, and actin, or we might just have to memorize it. But that's a weird thing that they have to do. And then they have things that are usually smaller functions. And so the example that's on this is the villi that's in your intestines. So you have your intestines, and sort of has all these little projections. And so what does that do? It increases the surface area, so then it increases absorption. And so then you can absorb the nutrients that you need. Little side story, my husband is a celiac, and whenever he eats gluten, it sloughs off the microvilli inside of his intestinal tract. And so when he was little, and when people that have celiac are little, they don't absorb the nutrients that they need, so they're oftentimes underweight. And so it's one of the early symptoms of having celiac disease, or being gluten intolerant. And so just a picture. And then some cells, some organisms, protists, they like scooch out some of their cytoplasm and cell membrane. So pseudo means fake. Pod is like a podiatrist, and so it's like a fake foot, and so it like scooches it out, and then like grips, and then like pulls the rest of it along. And so it's a movement thing, and sort of how they sort of jet that out and then sort of retract up back to it. That's done using microfilaments, so just along the edge of the cell. And so then cytoplasmic streaming is just exactly what it sounds like. It makes it so that the cytoplasm is moving around, and so it's not stagnant, and so then the gases and all the other stuff can be exchanged, and different stuff like that. And then we're going to talk about extracellular matrix, and then a few other things. So the cell wall is a rigid outer part of certain things. Plants is the easy one to remember. Some bacteria, some fungi, they have, and some protists have cell walls. We do not. And so what it usually has something to do with is structure and controlling water in and out. And so then this is just talking about different layers. It's just vocabulary. And then the bottom of the part is plasmodesmata. And so we have cells that are up against each other. And if they have a rigid cell wall, they sort of can get stuck against each other, but they might still need to exchange materials or different fluids or different things like that. There's little channels that connect them, and those are called plasmodesmata. And so that's just what they look like. And then this is just talking more about just extracellular matrix. So in general, extracellular, so it's outside of the cell, this matrix that sort of hooks things together, makes it so cells can talk to each other, gives a little bit of structure and different things like that. It's kind of a generalized term for things on the outside of the cell that have something to do with hooking stuff together. And so just a picture that shows those different things. And so then there's four big things that the different things on the outside of the cell do. One of them is support, and so if this cell's connected to this cell, then that is a supportive thing rather than sort of go blah. Adhesion means this is sticking to that. Movement is sometimes things need to shift next to each other, sometimes they don't, and so the extracellular matrix helps with that. And then regulation is sometimes has to do with identification of what kind of cell it is, should the hormones affect that cell or not, or different things like that. And so then this is talking about plasmodesmata again. Gap junctions are the same thing except in an animal cell. And then tight junctions and desmosomes, I'll show you a picture and hopefully that'll help you understand just where they are and sort of what they do. And so this I talked about is just a little, like a little tunnel, and then just a picture. And then a little bit about the other three, and it's still talking about the gap junctions are with animals. It doesn't specifically say that, but it is. Well, it says it at the top, but you probably didn't pay attention to that. Alrighty, just a picture. And then chapter 7, so this is about the membrane. And so our membranes are selectively permeable. Our phospholipids have a polar part and a non-polar part, a hydrophilic part, a hydrophobic part. When a molecule has two different things like that, it's kind of wonky. They're called amphipathic, and our phospholipids are amphipathic. The fluid mosaic model is, I like to think of it as a pool, and we put a whole bunch of ping pong balls, and so they sort of float on the top, and then we might put in some pool noodles and maybe like a inner tube, and so we have all this stuff. And so it's fluid and it kind of can move around, but if we sort of were to like push the surface of the water, all that stuff would shift around, and so the picture, the mosaic, would be ever changing. And so you can tell this was named in like the 60s, 70s time frame because it's kind of a groovy name. And so the picture's constantly changing. And so just a picture of the different parts. One of the things to point out on this, we talked about it a little bit in Unit 1, is the tails on the phospholipid. And so one of them is saturated and one of them is unsaturated. So we know the one on the kinked one is unsaturated. It has that double bond, and so then it has a little kink in it. That'll be relevant in a bit. And so this is just talking about proteins that are in there. We'll talk about why those are there in a sec. And then this is talks about sort of the movement of the phospholipids. Usually they sort of shift in and around each other. Like if they're like this, they're like shifting. Sometimes they go like whoop over like that. Not very much. But it happens, but not very often. And then the other things is it talks about fluidity and viscosity. If you remember from chemistry, the more viscous something is, the thicker it is. And so we want fluidity when it comes to the cell membrane. We want it to sort of flow and everything's sort of moving nicely, not rigid. And so there's two big things that increase membrane fluidity. One of them is the kink in one of the phospholipid tails. The other one is cholesterol. And so we sort of jam cholesterol in there between their two things. And so instead of being like this and trying to be all fluid, they're like a little bit separate, and then they can be more fluid. And so this is just an example, this is just an experiment that scientists did a long time ago that gave us evidence that the plasma membrane has a fluid behavior, and so all the pieces sort of move around and mesh. And then this talks about, along with the rigidity, because of like if they were packed in there together, one of the things that messes with membranes is temperature. And so if it's too high, it's bad. If it's too cold, it's bad, and it's worse. And so it makes it so that they get brittle and rigid, and then they can't fulfill their function. And so this is just the same picture that I showed you before. And so this is talking about how when we have temperatures, it affects the behavior, it affects the proper function of this plasma membrane. And then this is just the same picture again. And then now we're moving into proteins within the membrane. So this is a really, really busy picture, but if we sort of take a second and we look at it, we have different big proteins that sort of go across. There's some stuck on the outside. There are some that have the little geometric stuff stuck to it. And then we look, and where it says glycolipid, the little geometric stuff is stuck to the lipid part. And so glyco is like glucose, and that's like a sugar, which is a carb. And so if it is a carb stuck to a protein, we call it a glycoprotein. If it is a carb stuck to a lipid, we call it a glycolipid. And they have different functions. And so we'll talk through those, but just sort of take a little gander at this picture. And then, so the ones that are through, so they're integrated through the membrane, they're called integral proteins. And then the ones that are sort of on the outside, just like your peripheral vision is like the edges, those are called peripheral proteins. And so just a picture. And then proteins that are within and on the outside of our cell membrane have a lot of different functions. These are the ones that I want you to focus on. One is transport. And so the ones that are integral proteins, they make a little tunnel that things that are polar or things that are hydrophilic can use to pass through and get through that lipid part. Enzymatic activity, there's a whole bunch of different chemical reactions that happen in our body, and they don't happen with every single cell. And so there's stuff on the outside of excuse me, the cell that so that enzymes work or maybe don't work because they're supposed to or not supposed to. It's sort of a it's like an on-off switch. Sorry, I needed a little drink. Signal transduction, we're going to do a whole unit on that. But that is if this cell needs to make a certain sort of product and something on the outside has to come along to sort of start that product, we have to get a signal. And so it comes from the outside, it gets traveled down through, and then we make like a protein or an enzyme or something inside the cell. Cell-to-cell recognition, and so we're supposed to be next to each other, or maybe we're supposed to be in our bodies. Cell-to-cell recognition is also used in our immune system. It's when our white blood cells identify something that is not supposed to be in our body, and then they destroy it. And so, or maybe then intracellular joining and then attachment and all that I talked a little bit about when we talked about the extracellular matrix. And so it's when we have to stick stuff together. It gives us sort of like structural integrity between our cells and hooking it to different things inside of our body. So this is just a little picture of each. And this is just talking about the glycolipids and glycoproteins. I talked about this when we talked about the picture. There's the bell. And so our cell membrane usually is really busy on the outside and only a little bit of stuff on the inside. It's called sided. And so there's stuff on the outside of it because it needs to be on the outside for recognition. Okay, so we'll continue. Had some announcements there. And so next one. And so this is just talking about how things get into and out of. And so I talked a little bit about this earlier as well. If it can get across the lipid, it goes through relatively easily. If it cannot go through the lipid, it needs a helper. And so we have things called transport proteins. And so they transport things through the membrane. One thing that you might say, well, if that's lipid and water's super important, and oh my gosh, we should probably get that in and out. You're right. And there's a special little one called an aquaporin that does just that. And then we have carrier proteins, which are similar to channel proteins, except they sort of change shape, sort of like a little trapdoor. And then diffusion is moving from an area of high concentration to low concentration, no matter what it is. And then this is just shows a thing about things that are moving. So maybe it's one thing, maybe it's two things, maybe it's a bunch of things, maybe it's salt and sugar and fats and enzymes and hormones and all this stuff in our body. It just happens, but they're all sort of individual things, and they can figure out, like, is it high over there and low over here? Which direction should we go? Just the bigger picture. And then one of the things that we'll talk about a lot when we talk about ATP synthesis. Okay. So when we talk about ATP synthesis, when we talk about photosynthesis, we're going to talk about building up a gradient. And so when there is a difference between this side of a membrane and this side of a membrane, or the concentration over there and the concentration over here, that's a gradient. And so we'll talk more about that, but that just means that there's a difference. The bigger the difference, the faster stuff happens. But once it reaches down to being level, that's called equilibrium. But diffusion happens, it's called passive transport. There is no energy involved, it just happens. Just some pictures. And then osmosis is specifically the diffusion from an area of high concentration to an area of low concentration of water. And so oftentimes the salts and the sugars and all the other stuff might not be able to go across the membrane, but water can. And so we use water to affect the environment. And so this little tube, if we just take a closer look at this, there is at the top, the sort of top left U-tube, there's more solute on the right-hand side of that tube than there is on the left. And so if we look at that in terms of water, there's more water on the left-hand side than there is on the right-hand side. So then it's going to go from an area of high concentration to low concentration of water. And so then the water would scooch from the left-hand side to the right-hand side. So if you look at the right-hand U-tube, the water level goes up, but then the concentration of the solute is equal on both sides because we messed with the water, which then fixed the concentration of the solute. And so then we talked about tonicity. I drew some pictures on the board. We did a lab about the potatoes and the sugar solutions, and we figured out where it crossed that zero or isotonic level, so we can figure out the concentration of sugar inside of our little potato pieces. And so that's called the sort of the environment, the movement of water back and forth. It's called tonicity. And then we use words to describe the solution where the cell might be in. So it might be blood, it could be other stuff. That's just when we talk about a solution, we're sort of talking about the environment that the cell is in. So isotonic means that the solute concentration inside and outside of the cell is equal. Hypertonic means that the solute concentration in the solution is higher. And then hypotonic is the concentration of solute in the solution is lower. And so then these pictures sort of give you a visual as to what that means in terms of cells and the environment that you put them in. And so if we put an animal cell in a hypotonic solution, the environment that it's in, the water that it's in, is lower in solute. So it's higher in water. And so if it's higher in water outside of the cell than it is inside, and then that means that the water is going to go from an area of high concentration to an area of low concentration. So it goes into the cell, and so then it swells up. In an animal cell, we do not have a cell wall to protect us, so our cells burst. And then plants do, so they get like sort of bulgy. And then if the opposite of that is hypertonic, and so hypertonic is more solute on the outside of the cell, and so then that would be less water. And so then inside the cell would have more water, and so high concentration, low concentration would come out of the cell. So water would leave the cell, and so then the cell would shrivel up. If you've ever been in the hospital and you've seen maybe on an IV bag, it says isotonic saline solution, which means this bag has the right amount of salt, the right amount of water, so that your cells are nice and plumpy, nice and healthy. Not too plumpy, not too not plumpy, just perfect. And then osmoregulation is how our bodies control the amount of water in our bodies. So it's sort of how we get rid of or gain water to reach equilibrium. And so paramecium do it in a different way. Sometimes they're like, or we need some more water. Paramecium and protists are cool. And then this is just some vocabulary about it being too full or not full enough. It was also on the slide with the pictures. And then this is just another vocabulary word. Again, if you want a picture version, just flip back a couple slides and it will give you a little visual as to what that is, but just vocab. And then facilitated diffusion means it's moving from an area of high concentration to low concentration. There is no energy being used, but it needs a little helper. It needs facilitated. And so this is when we can't get across the lipid bilayer, so we need a protein to make a little tunnel. And so facilitated diffusion always has to do with proteins embedded, integrated, or integral proteins within the membrane. And so this just shows a picture of a channel protein and then a carrier protein. One's like a tunnel, one sort of like a little trapdoor. And then this is just talking about shape changes. And then just an example, a specific one. And then this is talking about that it's passive because just because it's facilitated doesn't mean it requires energy. Diffusion is diffusion, high concentration, low concentration, passive. And then active transport means that it moves from an area of low concentration to an area of high concentration. So it's going against the gradient. And so when you do something that just doesn't naturally happen, you have to expend energy. So active transport means that ATP is necessary. And so one of the things that does this is the sodium-potassium pump. If you took maybe psych, but anatomy, you may have talked about the nervous system and talking about like a nerve impulse and like the twitch of a muscle, and you have to build up a sort of coiling some up so that it can fire. The sodium-potassium pump is one of those things. So it requires energy to be expended in order to get the power to do that work. And so this just shows how we trade three sodiums, 2 potassiums, and so it's a little pump. And so it also gives you sort of the indication that energy is going to be expended because we're trying to build up a gradient. We're trying to sort of shove a bunch of things into a certain part of the environment so that it's sort of charged up and ready for our nervous system to work. And then this just shows sort of an overall thing. And then this membrane potential is associated with active transport with sodium potassium pump, and it is sort of it's an electrical way to describe the concentration gradient. Like when I said earlier, I said if the concentration gradient is big, then diffusion happens quickly, and as it sort of approaches equilibrium, the rate of diffusion slows. And so this is sort of associated with that, in that it's sort of our measurement of sort of power. And then this just talks about sort of the same sort of things. Just be aware of the words. Don't stress too much about what they are. Just so that when I talk about gradients later in the units, you'll understand. Same thing. Just vocabulary. And then just a visual that goes along with that. And so we're building up a gradient, which means that we're going to take some energy. This happens in cellular respiration. This happens in photosynthesis. It happens in the sodium-potassium pump. Just some examples we'll talk about as we go through the school year. And then sometimes we need a little helper. It's exactly what it sounds like. And so then we have little co-transporter. We have a little helper molecule that for some reason in certain processes, we just need this little extra thing. And so this is just an example of that. And then sometimes we need to get big stuff across. And so we have some vocabulary that goes with that. And so exocytosis means that we are going to get something out of the cell. So it's might be waste, might be something that we made inside the cell. So exocytosis means that maybe we made something in the membranes, and then we made a little vesicle, and it needs to get over to the cell membrane, and then it like makes a little, ooh, got my fingers in the camera, makes a little opening, then squirts the stuff out. So like exits the cell. Endo is the opposite, and so it's bringing stuff in. There's 3 vocab words that go with this that I want you to remember. Phagocytosis is cellular eating, so it's bringing in solids. Pinocytosis is drinking, so it's bringing in liquids. And then receptor-mediated endocytosis is we need to bring stuff into the cell, but we need to make sure it's supposed to come in. So we need a little receptor, a little guard out there for thing to be like, hey, I'm supposed to come in, and then he's like, okay, let's go, rather than like, hey, everybody come on in. So receptor mediated is a controlled entry of whatever it might be. And so this is just talking about the same thing. Some pictures to go along with that. And then this is just the same. This is drinking. Same thing, little pictures to go with that. And then this is talking about the receptor. The little thing, sort of the, I'm the guard that is making sure that you're supposed to come in here. This is called a receptor, and then a little molecule, it's called a ligand, sort of goes in and it fits in there, and then it's like, choot, and then it opens the door. So that's just the little thing that says, hey, let us in. That's called a ligand. And then this is just the same thing. The cool thing about receptor mediated is then it sort of envelops itself, and then the receptors that were on the outside are now on the inside. And so then our body doesn't, when it's inside the cell, it doesn't have these weird things sort of sticking out to trigger any sort of immune response that it, like, it's not supposed to be there. So it sort of like gets this like cloak around it that plasma membrane looks exactly like every other plasma membrane. And then this is just some pictures of the stuff we talked about. Same thing, just some pictures.